thank you very much for inviting me. It's really lovely to be here and see so many of you actually coming and participating in a meeting like this, because obviously you're the patient voice, so we need to be listening to you. So really you should be up here talking to me, but anyhow. So as Susan rightly said, we have this consortium which we call CLIC, so it's Cutaneous Lymphoma International Consortium, and the idea is that we do studies together, so international studies. So really historically, a lot of our studies were either single centre or maybe European or US, but we didn't all get together through the continents and work together, which we've now started doing. The other person we should mention here, obviously Maarten's here with me, is Yong Kim from Stanford, because she's also been really fundamental to this work. So she's an important person to know. And really both our institutions, Stanford and University Hospital Birmingham, for supporting the infrastructure for the database to allow us to do such studies. So I'm going to talk to you a little bit about a study called PROCLIPI, and it's an acronym. So PRO stands for prospective, means looking forward, and CLIPI is Cutaneous Lymphoma International Prognostic Index. So what are prognostic factors or markers? Poor prognostic markers are features that are associated with a worse outcome. And if we can identify patients who may do worse, then we can tailor their treatments to try and improve their quality of life. But it also allows us to pick out the patients who may do well anyway, who don't need toxic therapy, and make sure that we're not poisoning them with therapies that they don't need. So using those prognostic markers or factors, we developed something called a Prognostic Index. And this is a formula, a mathematical formula, that you can put in the poor prognostic factors, and it will come out with an idea of the likelihood of a good or a bad outcome. So this is what we're trying to do. At the moment, we just use the stage, but we know that's not that helpful. We know that some patients with stage 1 disease go on to get stage 2, stage 3, stage 4, and some patients with stage 3 or stage 4 disease do very well. So this is really to try and sort those patients out and help them so that they get the best possible treatment. And it's not an unusual thing. PIs are used a lot in other lymphoma. So would it be useful in mycosis fungoides? So it is, because there's a wide range of outcomes, and most people live a long time with their disease. So you need to make sure that your treatments are making life better, not worse. So the aim is to pick out those patients who might need aggressive chemotherapy or transplants, as we were hearing earlier from one of the patients. And also because you're living with your disease, to make sure that the quality of life is good. So we're not the first group to have this idea. This has been something which groups have tried to do for a long time, but it tends to have been single centre. So Madeleine Duvik's group in 1999 published the first index. But the most important thing with these indexes is they need to be international, because there's too much variation from single centres. The one which we published from London realised that patients with early stage disease have slightly different factors that are important than patients with advanced stages. And we divide that up so that patients with early stage have 1A, 1B or 2A, and patients with advanced have 2B, 3 and 4. So the first thing we did before we launched into our PROCLIPI was to go back and look at our patients who had previously been with us and their stages, and to see if we could get a few ideas of what may be important. So we chose 11 things listed here, like age and gender and how much blood involvement there was, how much skin involvement there was, and some specific markers too. And we had a tremendous effort. So we collected 1,275 patients. So, I mean, this is a big number, all patients 2B and above. So it's a huge number. And these are all our colleagues who worked very hard together with us to collect this data. And these, the ones in green, were the ones that we found looking worldwide that seemed to be important. So being older when you were diagnosed, more than 60. This is a marker in the blood. It's called LDH, and it's raised in some lymphomas. This is stage 4, which can be blood or node involvement. And this is something called large cell transformation, which, again, someone was talking about. And it's where when the normal size MF cells, which are sort of a medium size, get bigger. So classically 4 times bigger. So they look like sort of monster cells. And this was published in the Journal of Clinical Oncology, and it was very heartwarming to see sort of 77 authors from 24 different countries. So just a few slides on the prospective study, so the PROCLIPI that we're doing now. As I mentioned, we're doing an advanced stage and an early stage, so there'll be 2 different PIs to look at. And in Europe, we opened in July 2015, and the data is put onto a secure website. So each patient is consented, and then you have these buttons along the top to enter the data. And then once the data's entered, you save it, and then if they have another visit, you can enter the data for the next visit. It's a very bit small, but one of the most important things we're collecting, I think, for patients is treatments and treatment responses. Typically, we have very little studies on most treatments, particularly in early stage disease. I'm sure many of you get given steroids or phototherapy, but there are no large studies, so collecting this data prospectively is going to be really important. And these are our centres, even more than in the retrospective study. And in the first year, we've collected 432 patients. So, patients really want to be involved in this study, and they can see the benefit of it. So these are the recruitment from all of our centres. And these are the stages of patients. So 77% have early stage disease and 23% advanced stage. As I say, we call the early stage 1A, 1B, and 2A. And I don't know how much you know about the staging system, so I just put up a few slides. So when we talk about 1A, that's when you have just a small amount of your skin involved, and we have this random cut-off of 10%, where 1% is about as big as your hand. So if you imagine, that's 10%. So that's the 1A. And then the 1B is more than 10%, so more than sort of 10 handfuls. And then 2A is where the lymph nodes are enlarged, but they're not enlarged with lymphoma cells. And then we move on to the tumour stage, where the plaques become thickened and sometimes they bleed and become sore and weepy. Or the stage 3, which is when your skin goes red all over. And then the stage 4, where you either have these big cells. These are lymphoma cells. You're looking at high power. So these are red blood cells, and then these are the lymphoma cells in the blood, and they shouldn't be there. And that's a stage 4. And the other stage 4 is when there's lymph nodes, and these are lymphoma cells in the lymph node. So just before I finish, as I said, I thought treatment was exciting, so I just put this one up. So this is our early stage patients, and so far we have treatment captured on 229 of them. And the most common treatment was phototherapy, which was given as the first treatment in 32%. And the response rate was 77%, so good response. And that's people who were cleared by at least 50%. So it's not everything gone, but it's a good improvement. And then topical steroids, the next most common, in about 30%. And the response rate was about 63%. So again, a good response rate. So showing that, I guess, patients wouldn't use them if they didn't work, or you wouldn't keep coming to your phototherapy. And what was quite interesting is the next most common treatment was actually retinoids, which isn't necessarily what treatment we would imagine we would be putting patients with early stage disease on first line. Normally we would think that would be later. These data are probably slightly skewed because most of our centres are large centres where we see difficult cases because they patients want to come to see us if they have disease which isn't being well controlled somewhere else. So this may alter this. But overall, most of our treatments worked in about 2/3. So thank you very much. These are all our sites and trying to work together to get better research. And these are our sponsors who, without any of this and without any help, we obviously couldn't afford to do these studies. So we're always very grateful for having any support. And with this study, we've had a lot of support. So thank you. So I'm going to... I'm going to pass you over to Maarten Vermeer. Susan's already introduced him, and he's in charge of our federated biobank.
A very short introduction of myself. My name is Maarten Vermeer. I work as a dermatologist. I've been working on cutaneous lymphoma for the last 25 years, first in Amsterdam, later in Leiden, and we have a relatively large center and serve as the sort of national referral point for the diagnosing and care of cutaneous lymphoma in the Netherlands. Okay, let's skip that. So, about cutaneous lymphoma. First of all, the first thing we're interested in when we see patients that are, where we think we are dealing with a cutaneous lymphoma is what is the specific diagnosis? Which type of cutaneous lymphoma are we dealing with? And the second one is how extensive is the disease? So the staging procedures that have just been shown by Julia are very important. And this gives us information on the prognosis of someone, and this will help us select the most appropriate treatment. And this is important because in different stages of disease, the survival is different. Whereas, let's say in stage 1, the survival is 100% of patients, or 90% are still alive, really after a long, long time. Whereas that's very different for stage 4 patients. They have a much poorer survival. And that influences, of course, your approach to therapy. Now there are some difficulties with the data I just showed you. These are derived from a cohort of patients, so from a very large number. And we'd like to improve the staging system and have more individualized information. And that would answer the questions that patients can have, and we have with them, is what can I expect in the coming years? To which treatment will the disease respond best? How great is the chance that the disease will recur? And if so, will it only recur in skin? Or is there also a chance that it will recur in extracutaneous sites, so somewhere else in the body? And then everyone is unique as we are sitting here, but the same is true for your tumor cells. Every tumor is unique too. What we'd like to know is what determines the behavior of these tumor cells, and what can we identify markers that will inform us and you on how these tumors will behave in the future. In short, how does this particular cancer work? Now I have to go back to 1953, And the first thing that will come to mind to many of you is, of course, the ascent of Everest. But people would argue something which was equally important was the discovery by Watson and Crick that DNA helix is the three-dimensional structure of your DNA. And someone that should be mentioned here also is Rosalind Franklin, yeah? You already... Okay, because she made the pictures that would allow them to make this deduction that it's really a double helix. And in their publication there is this very, very nice quote, we're one of the best in the literature, I think, that it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So what does this mean? It means that one particular DNA code, if you copy it, will lead to the same code. And this will lead to the same code. So within this sequence of your DNA bases, that's the basis for your proteins, and this will determine the phenotype. Phenotype being the behavior of your cells. So this is how it should work. You have your genes, that determines how your proteins work. These will take care of all kinds of cellular processes, and this will determine the behavior of your cell, called the phenotype. So genes determine cellular function and phenotype. This also means that genetic defects, so if your DNA breaks or if there's something wrong with it, this can lead to loss of your proteins, or excess of a particular protein, or a changed protein with new functions. And all these defects in these proteins together can lead to changes in cellular behavior. They start behaving bad. And combination of genetic defects can result, in the end, in a malignant phenotype. What this means is the development of cancer. So in the end, cancer is a genetic disease. Yes. And this is just to point out, without going into detail, there are all kind of different types of genetic defects and differences on RNA level and the end and signaling pathways. But the big scheme that I just showed you, that's the thing we should keep in mind. So it's a genetic disease, yes and no. Because it's also a epigenetic disease. Now we go to a different type of mechanisms. These are mechanisms responsible for differences in phenotype, but they do not affect the DNA code. So the DNA sequence is the same, but something else is taking place. And these type of mechanisms are very important during embryogenesis, because with one fertilized egg, in the end we will end up with a whole human being, and in every cell of your body the DNA is the same, but your cell in your, let's say in your brain, is very different from your skin or your colon. So how does that take place? So differentiation and functional polarization. This is just an example. This is the switching on and off of different proteins will allow the development of different hematopoietic cells from out of one stem cell. So in your bone marrow you have one stem cell, but out of this one particular stem cell, using the same DNA, we have all type of different cells in our blood. And that's done by all kind of epigenetic changes. Now this sounds a bit funny, but if you walk through a forest, especially in autumn, it's a great time for epigenetics. You know why? Because all kind of epigenetic changes are taking place. In this bear who is hibernating, it's changing its metabolism. All kind of genes are switched on and switched off to allow it to sleep for a very, very long time, to have a very, very low metabolic state. And this is maybe the best example. This is a caterpillar, very nice phenotype. In this cocon, I think it's called. Yeah. Next time you walk through the forest and you see it, and you're walking there with your child or your grandchild or whoever, and what's going on? He said, well, there's epigenetic reprogram going on, because next thing, using the same DNA, I mean, the DNA hasn't changed, now you suddenly have a caterpillar. Oh, no, a butterfly. No. Okay, so you got the idea. So how does this work? It works with access to DNA. So we have the same DNA code, but if the whole transcription machinery has access to it and can translate the code into protein, is determined by two processes. By chemical modifications of your DNA itself and chemical processes of the proteins where the DNA is wound around. And this will lead to a condensed or a relaxed state of your nucleosomes, and here in this state there's access to DNA, and now it is turned on. So epigenetics are the switches for protein expression. So this is some theoretical background, and now we understand, using genetics and epigenetic changes, a healthy cell can turn into a cancer cell. So we can recognize this under the microscope. This is a cancer cell, and this is what has gone wrong. So the challenge is, can we find genetic and epigenetic markers, biomarkers, that give us more information on what we can expect in the coming years from this particular cancer in this particular patient. And then the CLIC study is a very, very nice vehicle or framework to answer these questions, because it will, in the end, it will result in a very well-defined group of patients, and the cohort of patients included in the study will give us an excellent opportunity, I think, to try to answer these questions. So the purpose of the biobank is to develop a framework to do just that. And the goal of these studies will be to identify the biomarkers that will give us clinical information on prognosis and better therapy. Design is as such, participating centers will register their patients in this database, that was just pointed out, but they will also notice if biomaterial, so skin biopsies or blood, has been stored. And the idea is to construct a virtual biobank. So all the material will stay on site, and this is done on purpose because it saves us a lot of legal issues. So there's nothing strange happening, so just the normal material that has been collected during the diagnostic process, which stays in the institute, is now registered in the biobank, so you just know it's there. Of course, we have to have a very careful process on what type of studies we will be doing. So proposals will be submitted to a steering committee. They will select the most appropriate study. We will then identify the material that will be useful for this particular study, and then the collaborating centers will be asked if they are willing to collaborate to do that. Opportunities are the translational studies that I just described will profit from the design of the CLIC study. So prospectively, so longitudinal in next couple of years, in different centers all over the world, the detailed clinical phenotype. And a lot of work is currently being done on all kind of genetic markers. So I think we will have a lot of candidate genes and candidate defects that we can test in our studies. And this is just an example to show you that this kind of approach can work. This is a whole range of biomarkers, both on the DNA level, on RNA level, and on protein level, that had been described for Sezary Syndrome. No one was used them. They had just been described and they were just out there. And what we did, and it's difficult to do studies in this particular disease because it's quite rare, but we had a collaborative effort led from our institute in Leiden, but together with the people in London and Paris and Mannheim, that was Finland, and this is Mannheim coordinating the German centers, and then Turin. And from all these centers we got a lot of specimens, and this went through quite a complicated workflow, isolating all blood and cells from blood, the DNA, the RNA, and cells themselves. And in the end, this was quite successful because we just had a number of publications showing the diagnostic value of these novel biomarkers. We identified a new prognostic marker, and we are now trying to use these diagnostic markers in early stages of disease. So really trying to find out if people come to our clinic where we cannot make the diagnosis with the current diagnostic tools that we have, maybe these markers can help us. So the PROCLIPI was just introduced. We already have a lot of patients entered in the database, and we hope to embark on these type of studies pretty soon. And in the end, I'd like to acknowledge, of course, the SPAS Foundation, the Cutaneous Lymphoma Foundation, and I firmly believe that friendship and collaboration with our patients and colleagues will help us to solve the questions that I just showed you. Thank you, and I'm ready to take questions.
My doctor's Red Sloan Kettering. I'm MF stage 1A, 5 years now. I have donated my cells, blood tests, skin biopsies. How do I know if the International Consortium will get these cells? I can't, or data? I mean, where do they go?
Yeah, it's a very good question, actually. And most of our research, we would have in the patient information that if they want to know what happens to their biopsies and where it's published, that they do get access to that. So if you spoke to your doctors, they would be able to know which studies your samples will be used for, but it would have to be from your centre because any data we got would be completely anonymised, so we wouldn't know who you were. We might know your type of disease, but that would be all.
That's true.
Right.
It's a very important question, actually, and this is true, as Julia pointed out. This is true for all over the world. So everywhere we need to have a lot of legal issues have to be solved because we don't want we do not want a situation that patients do not know that they're taking part in these studies, or data or things will come out and then we suddenly realize that patients have never been informed that they in fact are participating in this. So we've been strongly stressing that to all the individual centers, that they should inform their patients, etc., etc. And in fact, this is something that has a lot of attention now, and all the medical journals don't even want to accept publications anymore if you cannot show them that you have informed your patients and that they know that they are taking part in these type of studies, which is a good thing. Yeah, and normally it is normally it's the case. You can always ask: is my material, are my biopsies, or are my blood cells, are they being used in studies? And any time I think in the Netherlands at least, and probably in all over the world, any time that you think, well, I'm not satisfied with it, I want to stop my cooperation. You, as a patient, you are the owner of the material.
Right.
This is a direct follow-up to Fran's question. I'm 1A and was diagnosed early in 2013, and I just want to know, how do you sign up to do this, or does your doctor ask you? Do they solicit you? Because this is actually the first I've heard of it, and I would be absolutely delighted to do this because I can't give blood anymore, I can't be an organ donor anymore, maybe my corneas, but maybe not. So at least I could do that.
Which centre are you?
Well, my doctor is Dr. Geskin. I'm really lucky.
Okay.
And she's at Columbia Presbyterian.
Well, if you spoke to Larissa Geskin, then she'd be able to give that information. It has to be done, as I say, on a centre by centre agreement because of patient confidentiality. There's only a certain amount of data she can look at, but she's definitely on board and she wants to join in. So a bit of patient pressure is perfect.
Are you still recruiting from the United States, different facilities and hospitals? Because it seemed like there was only one in Stanford at this time.
The legal process of getting our US sites open is always a little bit tougher. As you'd have seen from the retrospective study, we had 7 or 8 US sites involved, and we would imagine the same sort of number would join the prospective. We are an inclusive consortium and an inclusive study, so we don't want to push centres out. We want everyone to join in, but that means that they have to have some sort of integrity of the data that they're providing us to make sure it's correct, because there's no point putting a lot of data in that actually doesn't make sense, because then we don't get anything sensible out at the end. So we want those centres who are used to doing clinical trials and have a large patient base to join us in this attempt, because this way we should get good quality data and hopefully make a difference to our patients.
A question from online. In Sezary Syndrome, after a genomic analysis of a patient's blood has been conducted, what question should the patient ask about the results? For example, is there a match to a current treatment? Are there prognostic markers in terms of outcome? What else?
It's a very good and a pretty difficult question to answer. I think there will be genetic markers that help us to sort of get a feel for what's the most likely clinical course, but although a lot of different genetic defects have been identified, very small ones like point mutation, but also big ones where whole chunks of DNA got lost or got duplicated, it's still not the case that a specific alteration in the genetic material, it correlates with with prognosis. And we very recently published that the expression of T-plastin, which is a protein that is normally not expressed by T cells, when it is expressed, it is associated with an adverse prognosis. So this is it. This is just out. This has just been accepted. This was one of the things that came out of the collaborative study. The difficulty with that is, and we also put this in the study, that this is one single observation made on 60 patients. And in fact, what I would like to do, we're trying to keep the consortium together, and in the next couple of years, we'd like to see if this observation holds up, if you would look at it in newly diagnosed patients, and then see in the next couple of years how the disease develops. Yeah.
I'll go back here. Hang on. I'll go here and then come over there.
This may not be the field inappropriate question, but I've been doing a lot of reading on bioactive compounds in foods that modify the epigenetic behaviors. Are you able to comment on any studies or relevant information?
Not really. as you were saying, you've been reading a lot about it, so it might be that you know much more about it than I do. What I know is it's a very dynamic field at the moment, and the interesting thing is epigenetics has become a sort of an interface between what happens in your genes and what happens in your environment. And part of your environment is the metabolism, so the metabolic state you're in. Part of it is the activities that you do, the UV exposure that we have, the exposure to different types of food, including foods that seem likely to directly have influence on epigenetic markers. But they're still not. It's not like, oh, if I eat this or I do not eat that, then this epigenetic switch will go this way or that way, and that will lead to this change in the behavior of T cells or tumor cells. A sort of more generic statement, which I think is true. A healthy diet is always a good idea.
I just wondered if any of your charts and information was available online, like the different treatments that people had received.
Yeah. So the paper which we talked about in the Journal of Clinical Oncology, that's all available. You can download that. There's no problem. Or I can give a copy to Susan and she can distribute it. We have done a similar treatment paper, which will also be available shortly, but isn't at the moment. This data, because it's preliminary data and it's not written up, we can't make it available. But as soon as it's published, then absolutely it'll be there for everyone. But we have to be a little bit careful about giving out data when it's not published.
And that's the really wonderful thing about being so connected in all the different, we're connected with the clinical community, so as all these things are happening and occurring, and we have people that come and talk to us, it's extraordinary. And I can say that in a lot of other diseases, we don't have this kind of collaborative, connected, so we're really lucky. We're really, really lucky.
Hi, in your testing, have you found a high preponderance that the patients have developed cutaneous T cell as a secondary cancer and have had earlier in their lifetimes different types of cancers, what types of cancers, and I'll just be curious.
Yeah.
So.
Thank you. It's really an excellent question. One of the things, the basis for this is that there are some defined genetic defects, and that are hereditary, so it runs into families, and this leads to that members of these families that have that defect have a higher likelihood of developing particular types of cancer, like the BRCA1 defect will predispose women for the development of breast cancer. Till now, there's not a firm correlation between cutaneous lymphomas and one of these known familiar cancers. It might be that because cutaneous lymphomas are so rare that such an association, in fact, it does exist, but it's very weak. And we've been looking a little bit into this, and I got the impression that quite a number of the women that we saw had a breast cancer earlier in their life, but it's a cancer that's quite common. So I'm not really sure if it's a higher chance or that this was just a chance observation.
I just wanted to know when you get the skin or the biological tissue, do you get a history of the person? Also, is it a like very all-encompassing history?
Absolutely.
Or
yeah. So
like exposure to toxins or whatever?
well, we get a complete data set, so we get clinical blood, lymph node data, and the federated biobank, which Maarten runs, is attached to that data. So the data is complete. It's anonymized. There is entry for previous malignancies, any unusual treatments that you've had before. So you can put some of these things in. Again, it relies on your physician knowing it and putting it in.
You talked about developing a mathematical model
Yes.
that would be predictive. And you kind of looked back on the history of a number of patients. But listening to some of the work we have on epigenetics, it sounds like those are the factors that we need to put into your model. So to finalize your model, Is it really a function of completing this research work?
Absolutely. Yeah.
Okay.
So there's no
That's-
reason why you can't have biomarkers in a prognostic index. There's no reason.
That's precisely the way forward. So in the end, you look at markers, then you see in some patients have them, some of them don't, and the first thing to do then is to correlate it with other things to see, are these the patients that did well, or were these the patients that developed more aggressive disease, or does it associate with large cell transformation, whatever.